Good morning, guys. Today I will participate with you the way how to answer in molecular biology and genetics. So we have right now the variant 1 or variant A with the first question that has been written here. Individual nucleotides in a nucleic acid molecule are linked with the correct answer is 3 to 5 phosphodiester found like individual nucleotides in a nucleic acid molecule. Second question, if the content of thiamine residue of the total number of residues of DNA is 20, then the content of guanine will be here that we have to answer according to the equation. So the base pair rule in DNA states that the total percentage of adenine A must be equal to the total percentage of thiamine T, and the total percentage of cytosine C must be also equal to the total percentage of the guanine G. So in other words, A equal T and G equal C as well. So if the content of thiamine residues in DNA is 20%, then the content of adenine will be also be 20% or percentage. So therefore the combined content of adenine and thiamine is 40. That's what we see like in number 1. So question number 3. Select everything that is most like characteristics of free RNA. The answer will be the based of option given. The most characteristics features of RNA are single chain, as well contains uracil, contains ribose, and contains disoskiribose. And just about the ribose, only like type tRNA contain modified from ribose called disoskiribose. As well, question number 4. The following enzymes are not involved in the replication processes. So the correct answer is number 4, DNase. Because it's break down the DNA by hydrolyzing phosphodiester bonds between nucleotides, and it's not involved in the replication processes, but instead plays a role in DNA degradations and recombinations. As well, on other hand, DNA polymerase is key enzyme involved in the DNA replication and responsible for catalyzing the polymerization of the nucleotides from to form new strands complementary to the template strand of the RNA. Yes. Question number 5. Name the substrate for the translation processes. We will see that the correct answer is amino acids and mRNA, because during the process translation, ribosomes reads the codons of mRNA and pairs them with the appropriate amino acid. So tRNA transfer RNA molecule bring to the amino acids to the ribosome. So the amino acids also added to the growing polypeptide chain. Question number 6. Operon is, the correct answer is number 1. It's a unit coordinated genetic expression. Yes. Question number 7. mRNA processing is, we will see what is. Here I must identify some other points. The option that correctly describe the mRNA processing is number 4, which is the fragmentation of the genes of the primary transcript. Actually, that's easy, and I had write some other information. You may return to it, and some other information cannot be right. Question number 8. Information about structure of protein is transmitted to the cytoplasm. The option that correctly describes the transmission of information about the structure of protein to the cytoplasm is number A, or number 1, messenger RNA. And as well has written the why we choose this one. Codon of initiation, a section of the chain that determines. So the correct answer is number 2, start of RNA transcription. Yes, and as well we mentioned about the codon of initiation, that's specific 3 nucleotides sequence, as well AUG that occurs near to the 5 end of the messenger mRNA, messenger RNA. Number 10, ribosomes in the process translation are combined into a structure called polysome. Yes, because in translation the process protein that's synthesis what's called, yes, process protein it's called the ribosomes, or the genetic information encoded in mRNA. Question number 11. The following can attach the aminosyl site of the ribosome during translation is tRNA, but except the annotator. Question number 12. Specify the sequence of the first 3 processes occurring at the initial stage of the eukaryotic ribosomal cycle elongation. It will be like the first aminosyl tRNA connected to FE1 and GTP joins the P site. Number 3 and as well number 2, which is methyl tRNA in the A site. As well, there is also aminosyl tRNA in A site. Okay. Question number 14. Specify the components required for translation. The correct answer is, first of all, functional ribosome, as well energy of substrates, amino acids, and as well GTP energy. As well in the initiations factor. Question number 15. As well, for who want to other information, like extra information, he may return to the extra information about this question. Question number 15. Specificity of substrate required for the transcription processes. As well, it will be triple phosphate NTPS, and the other information has been written in the description. Number 16. A gene is we will see that segment of DNA where information about the primary structure of the polypeptide is destroyed. Number 16. Process of recognizing is it will be number 4, as well, recognition and selection of amino acids. Spatial corresponds of nitrogens passes of each other nucleic acid molecule is carried out according to the principle. Which principle? Here I could tell that's the principle that describes the spatial corresponds of nitrogens bases to each other in nucleic acid molecule is complementary. so it will be number 2. Number 19, the theory of matrix synthesis was proposed by, it will be number 3, Nikolai Koltsov, who is Russian like biologist and cytologist. It was like in 1928. Question, okay. Number 21, the post-translations modification of a protein occurs in, here you may also guys pay attention about the post-translation modification of a protein occurs in lysosomes, so number 4. Number 22, the triplet of ATS nucleotides in a DNA molecule will correspond to the codon of the iRNA molecule. It is the UAG. So it's correct. Select correct. Answer is number 2. Select the features corresponding to the characteristics of protein metabolism in the human body. First of all, the breakdown of protein molecule to amino acids occur in cells. That the one answer is correct. As well, breakdown of protein molecule to amino acids occur in the digestive tract. That's also correct. And as well, the end products of decomposition are carbon dioxide, water, urea, as well is correct. The daily requirement is 100 to 150 gram, also correct. According to the option number 4 is incorrect because glucose and fatty acids are not the end product of protein breakdown. Yes, number 24 question, what properties are characteristics of translation? Here please pay attention about it. The option 1, occur in the cytoplasm, is incorrect because it describes transcription, not translation. That's the first one. So the synthesis of RNA or on a DNA template, that's incorrect. Option number 2, occur in the cytoplasm, is correct because translation occurs in the cytoplasm. Option number 3, is also incorrect because that describes DNA replication, not translation. So the duplication of DNA molecule is incorrect. Option number 4 is also correct because translation occurs in ribosomes. Option number 5 is incorrect because it's not clear what's meant by a matrix synthesis in this context, so it's not correct. Yes. Number 25, after the initiation of translation, a reaction occurs. It will be elongation of the polypeptide chain because it describes the next step in the process of translation after initiation. Number 26 question, ribosomal RNA is, we will see that's the correct option is number 2, structure directly related to the implementation of genetic information. Protein spelling is, the correct answer is number 5, cutting out part of amino acids. The internal self-removing part of post-translation protein, it will be the internal self-removing part. Post-translation protein is called intine. The beginning of mechanism protein selecting is number 2, translocation of N-terminal site. Protein following is number 1, specific protein conformation. We will stop here in number 30. and we will like